Assalamu alaikum dear students. Welcome to Dr. Roomi lectures. Today we will study another topic from endocrine physiology. Have you ever wondered what determines the metabolic state in our body during feeding and fasting? In the fed state, when we have taken meal and there is high level of blood glucose and amino acids and fatty acids in our blood, there is production of hormone insulin, and during fasting state we need to increase our blood glucose level and there is catabolism going on in the body, and the hormones for catabolism are different, and these hormones are glucagon, growth hormone, cortisol and epinephrine. Pancreas is a gland which has both exocrine and endocrine portions, and by weight the exocrine portion of pancreas is 98 to 99%. This is made by the acini of pancreas, and this creates watery, alkaline, enzyme rich secretion which is called as pancreatic secretion. Pancreatic secretion is one of the most abundant secretions by the GIT glands. This pancreatic secretion from the exocrine part enters through the ducts into the second part of duodenum. Second part of pancreas is the islets of Langerhans. It is just 1 to 2% of the pancreas by weight, and this secretes glucagon and insulin mainly. These islets of Langerhans are scattered throughout the pancreas, and they are about 1 million clusters of cells. Each islet of Langerhans contains three major types of cells. The most abundant cell in the islet of Langerhans are the beta cells, which are mainly present in the center or middle region of the islet. These cells secrete insulin hormone and also amylin. We know the functions of insulin, however, the functions of amylin hormone are not well understood. Alpha cells, which are present towards the rim of the islet of Langerhans or towards the periphery, these are just 25% of the total weight of pancreas, and these cells secrete glucagon hormone. Delta cells are just 10% and they are scattered in between the alpha and beta cells. These cells secrete somatostatin hormone. Another kind of cell, PP cell or polypeptide cell, also called as F cell, is just 1% and it secretes pancreatic polypeptide hormone. This pancreatic polypeptide hormone may have a possible role of reducing appetite and food intake. The rest of 4% mass of islet of Langerhans is made by connective tissue, blood vessels and nerves. What's unique about the blood supply of the islet of Langerhans? Blood first moves to the center of the islets, and in the center, because beta cells are abundant, so it picks insulin from there, and then blood moves towards the periphery where alpha cells are there which release glucagon. So this insulin which is coming from the center from the beta cells, it inhibits the secretion of glucagon hormone from the alpha cells which are present towards the rim of the islets of Langerhans. Different hormones in the pancreas control the secretion of each other, and this is possible because of the paracrine mechanism. The cells present in the pancreas, especially the alpha, beta and delta cells, they have gap junctions in between and they make a functional syncytium. Insulin from the beta cells inhibit the secretion of glucagon from the alpha cells, and glucagon from the alpha cells stimulate beta cells to increase the insulin secretion. And the somatostatin coming from the delta cells, it inhibits the secretion of both insulin and glucagon from alpha and beta cells. So this is because of paracrine mechanism. There are different type of messengers in our body. Those messengers which in one tissue are secreted from one kind of cells and then they affect the secretion of second or third type of cells in the same tissue are called as paracrine messengers, and this kind of control is called as paracrine control. Let's briefly talk about somatostatin hormone. This hormone is released from the delta cells of pancreas in response to the increase in blood glucose and amino acids, because after taking a meal when food is being digested, then these nutrients increase in our blood circulation. So this somatostatin hormone is like a brake which decreases the digestion and absorption of the nutrients, and hence it does not allow the excessive increase in the level of these nutrients in our blood. So it is a negative feedback mechanism. Moreover, somatostatin also decreases the excretion of insulin and glucagon from the alpha and beta cells through a paracrine mechanism. Somatostatin hormone also acts on the mucosa and the wall of GIT, where it decreases the blood flow of the intestine and also decreases the motility. Moreover, it also decreases the other hormone secretions from stomach and intestines. For example, it decreases gastrin secretion from stomach, cholecystokinin and vasoactive intestinal polypeptide secretion from the small intestine. Somatostatin hormone is also produced from hypothalamus, and this is also called as growth hormone inhibitory hormone, as it decreases secretion of growth hormone from anterior pituitary. Now let's talk about insulin hormone, which is synthesized from rough endoplasmic reticulum. Initially, insulin is in inactive form, which is called as proinsulin. Proteolysis of the proinsulin takes place in the Golgi apparatus, and it is converted into insulin and C-peptide. C-peptide is a connecting peptide which joins the two chains A and B chains of insulin hormone. So you can see here, when there is proteolysis of the proinsulin, then C-peptide or the connecting peptide is separated, and A and B chains, they are joined to each other by the disulfide bonds. Insulin is a small molecule which has only 51 amino acids. A chain contains 21 amino acids and B chain contains 30 amino acids. It's important to note that proinsulin is physiologically not active. It's not having the actions of insulin hormone. C-peptide is also not physiologically active, and when we separate the two chains of insulin hormone A and B chains, once again insulin is not able to perform its action. Now let's talk about the connecting peptide or C-peptide, which is part of the proinsulin hormone. However, when proinsulin is proteolyzed, then C-peptide is separated and only A and B chains are united together to make insulin hormone. This C-peptide is a small polypeptide which is having 31 amino acids. It's not having any activity of insulin. So it is physiologically not active, which means it cannot stimulate the receptors of insulin. Equimolar concentration of C-peptide and insulin is present in the secretory vesicles, which means that there is 1-to-1 ratio of C-peptide and insulin hormone within the secretory vesicles, which are made by the Golgi apparatus. The levels of C-peptide in the blood can be measured by using radioimmunoassay, and this is important to measure the endogenously produced insulin in our body, and this may be especially important in diabetic patient who is taking exogenous insulin. I hope you know that there are two types of diabetes, type 1 and type 2 diabetes. Exogenous insulin is the one which is given from outside of the body to a diabetic patient, and we don't give any C-peptide along with that. But the insulin which is endogenously produced within our body from the beta cells of pancreas, when one molecule of insulin is produced, one C-peptide molecule is also produced. So the level of C-peptides measured in our blood is directly proportional to the activity of beta cells. So it means if beta cells are more active, there will be more level of C-peptide in our blood, which means more endogenous insulin is being produced. Now I ask you a very interesting question. What do you think about the levels of C-peptide hormone in a type 1 diabetic patient? My options are very high level of C-peptide, normal level of C-peptide, very low level of C-peptide. What is your answer? The answer is, in type 1 diabetes mellitus, beta cells of pancreas have been destroyed by some autoimmune process. And because the beta cells are destroyed, so there is no insulin or very less insulin being produced in our body, which means that when we will measure the C-peptide level in our blood, it will be very low or it will be absent. Insulin hormone is stored as a hexamer crystalline complex within the beta cells of pancreas, and this is along with two zinc atoms. Calcium is very important electrolyte in our blood which is required for the release of insulin. Calcium is required by many hormones and neurotransmitters to be exocytosed from the cell. Once insulin is released, it will be transported in blood as a monomer, and this is transported in unbound form. Why? If you compare insulin with thyroid hormones, we know that thyroid hormones, they are transported in blood in bound form. The answer is that thyroid hormones, they are highly lipid soluble. So in order to transport lipid soluble hormones like thyroid hormones, steroid hormones, we need globulins and albumins. But because these are already protein hormones, so they are water soluble and can be easily transported in blood in unbound form. Half-life of insulin is just 6 minutes, and that insulin which is not bound to its receptors, for example on adipose tissue and muscle tissues, will be removed from our blood within 10 to 15 minutes by enzyme insulinase in the liver. So as compared to lipid soluble hormones, the half-life of water soluble hormones like insulin is very less, and also they are cleared from blood very quickly. So what is the significance of having a shorter half-life and smaller clearance time for insulin? It's because if our blood glucose level is already dropping, we don't need any more insulin, and the insulin which is already present, its action will be quickly finished because this insulin will be removed from our blood very quickly within 10 to 15 minutes. So let's suppose if insulin is there and it's performing its action for a longer period of time, your blood glucose level will keep on coming down, 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 and the person may go into hypoglycemia. Now let's talk about the mechanism of secretion of insulin from beta cell of pancreas. The most important stimulus for release of insulin from the beta cells is your blood glucose level after a meal. You take a meal, for example, roti, rice, that will be digested in our intestine, and when glucose is released, that glucose will come through the blood circulation on the beta cells of pancreas. On beta cells, we have GLUT2 transporter. This GLUT2 transporter, unlike GLUT4, is not insulin dependent, which means glucose can pass through it by facilitated diffusion and can enter into the cell. Once glucose is in the cell, it will be oxidized by glycolysis and Krebs cycle, and ATP will be formed. That ATP blocks potassium channels. You know that potassium is more abundant inside the cell as compared to outside. When you block these potassium channels, the diffusion of potassium from into outside will be decreased. So potassium being a positive ion, it will be accumulated inside the cell, and that will cause depolarization, which means inside of the cell will become more positive. This change in the membrane potential will open voltage gated calcium channels, and because calcium is more abundant in the extracellular fluid as compared to inside, through diffusion calcium will pass from out to inside of the cell, and this calcium will act as a trigger for these insulin vesicles. These vesicles, they will move towards the membrane, and by exocytosis insulin will be released. Some other factors which increase the release of insulin hormone from the beta cells are amino acids, especially arginine amino acid, which also increase in our blood circulation after taking a meal, especially the protein meal, and increased level of fatty acids, which increase after taking a fatty meal, and also intestinal hormones, for example gastric inhibitory peptide, also called as glucose dependent insulin entropic peptide, and glucagon as well, and growth hormone and cortisol hormone. You may be asked this question in examination that a patient A is given 75-gram load of glucose orally, and the other patient B is given 75 gram of glucose intravenously. So which out of these two patients, A and B, will have more insulin secretion? What is your answer? The answer is patient A will have more insulin release. Why so? It's because when you take glucose orally, that increases the production of some hormones from the intestine, especially this GIP hormone, which is also called as glucose dependent insulin tropic peptide or GIP peptide, and also the other hormones like GLP-1 hormone. These hormones increase the secretion of insulin after an oral load of glucose. Similarly, oral intake of amino acid will have more release of insulin as compared to the amino acids given intravenously. Why glucagon increases insulin secretion? Is because glucagon increases the blood glucose level, and when blood glucose level is high, then in order to enter that glucose into the cell so that it can be used, there is release of insulin hormone. Just like ATP can block potassium channels, a kind of anti-diabetic drugs, sulfonylureas, can also block the potassium channels, and their examples are glyburide, glipizide, glimepiride. And because by blocking the potassium channel they increase the release of insulin, that's why these drugs are also called as secretagogue drugs, which means they increase the secretion of insulin hormone from the beta cells of pancreas. What do you think? Can we use sulfonylurea drugs in a patient who is having type 1 diabetes mellitus? Yes or no? The answer is no. It's because in type 1 diabetes mellitus, the beta cells of pancreas, they are destroyed. And when the beta cells are not there, are very less in number, then sulfonylureas cannot increase the secretion process. These drugs, sulfonylureas, are only useful to increase the secretion of insulin in type 2 diabetic patient. So just to summarize, this blood glucose level is the most potent stimulus to increase release of insulin. And insulin secretion starts when blood glucose level becomes more than 80 grams per deciliter. And when this blood glucose level becomes more than 100 grams per deciliter, then the secretion of insulin is directly proportional to the blood glucose level. And the oral load of glucose will have more effect in the release of insulin as compared to the similar quantity of intravenous glucose. And the most important potentiating hormone in this case is GIP, which is glucose dependent insulin tropic peptide or gastric inhibitory peptide. Some other hormones which can also play a role in this case are gastrin, secretin, GLP, and cholecystokinin hormone, which means these hormones after taking a meal also are responsible to increase the secretion of insulin. but the most important potentiator is GIP hormone in releasing insulin after taking a meal. So in today's session, we have studied the histology of islets of Langerhans, the proportion of various kinds of cells and also their location and what do they secrete. We have also studied the chemistry of insulin, how it is transported in blood, its half-life, and its clearance from blood. Moreover, we have also studied the factors which increase and decrease the secretion of insulin hormone. In our next session, we will study the mechanism of action of insulin and also the actions of insulin on carbohydrate, lipids, and protein metabolism. So thank you so much for watching this video. See you next time with another video.